Look at your pepper plants right now, and reach down and pull one leaf toward you so you can see the underside. That pale, dusty-looking coating on the underside, or the dark spots beginning to show on the top, that is not bad luck. That is not a difficult summer. That is something you could have stopped for the price of a single 5-cent tablet. And nobody in the gardening aisle ever pointed you toward it, because there is nothing in it for them when you know. Here is the thing that will follow you through this whole video. There is a biological alarm system built into every pepper plant you have ever grown. It is real. It has been studied in peer-reviewed journals for over 45 years. When that system is switched on, the plant produces its own proteins that break down the cell walls of invading fungi, that stop bacterial wilt before it reaches the stem, that hold off gray mold and root rot and black mold all at once, not by killing the pathogen with chemicals, but by making the plant do the killing itself. The switch that turns this whole system on costs 5 cents. You can buy it at any pharmacy. The spray companies have known about this since 1979, and in over 45 years, not one of them has printed it on a label. Before you reach for another bottle this season, stop and think about what you have already spent. A bottle of chlorothalonil fungicide, the kind sold under that green label you have seen 100 times, runs around $14 to $19 per bottle, and you go through at least 2, sometimes 3 per season, because the instructions say to reapply every 7 to 10 days, and the season is long. Copper fungicide costs around $10 to $16 for a small bottle, and it does something different. So now you have 2 products. Add in a mancozeb-based spray for late-season blight protection, that is another $15 to $20. By the time August comes, you have spent $30 to $40 on fungicides alone, and your peppers still got sick in July, because every one of those products treats the surface of the plant, not the system inside it. You spray the leaf. The disease is already in the root. You spray the stem. The pathogen is already inside the crown. The spray never reaches the place where the problem lives. The aspirin path costs 40 cents for an entire 16-week season. 8 tablets at roughly 5 cents each, dissolved in water one at a time. 40 cents total, compared to $30 or $40 for the chemical route. That is not a small difference. That is 99% of your fungicide budget sitting untouched in your wallet, and a plant that is healthier than the chemically treated one because it built its own immunity instead of waiting to be rescued. So why has nobody told you this? That question deserves a real answer, not a comfortable one. The garden center does not tell you because the garden center sells spray. The gardening videos you find online are often funded by the same companies that make the spray. The agricultural extension bulletins were written in a decade when synthetic fungicides were the industry standard, and nobody has gone back to rewrite them. This is not a conspiracy. It is just commerce. Nobody makes money on a 5-cent aspirin tablet. Nobody builds a company on a recipe that requires nothing but a jug of water and a drugstore purchase. So the knowledge sat in academic journals, confirmed over and over, and never made it to the person who needed it. You, standing in your garden in July, watching your peppers fail for reasons you were never given. Stay with me through this, because the biology is the part that will change how you see every plant in your garden, and the protocol at the end will tell you exactly what to do tomorrow morning. Here is the picture that lives in most gardeners' memory from at least one bad summer. You come out to the garden on a Tuesday, and everything looks fine. By Thursday, there is a dark ring at the base of one plant, almost like a bruise on the stem right at the soil line. By Saturday, that ring has gone soft and the plant is starting to lean. You stake it You water less. You add fertilizer, thinking the plant is weak. By the following Wednesday, the whole stem has collapsed, and the pepper that was setting fruit is now lying on the ground with its roots exposed. And they are black. That is Phytophthora, root and crown rot, and it moved from first visible symptom to complete plant death in about 8 days. You pulled the plant, you threw it away, and you blamed the weather, or the soil, or the seed company, because nobody told you there was a window, in the weeks before that bruise appeared, when a 5-cent tablet dissolved in a gallon of water would have armed that plant against the exact pathogen that killed it. You are not to blame for not knowing this. The person who taught you to garden, whether it was a parent, a neighbor, a book, or a YouTube channel, was working with what was available to them, which was the same aisle of fungicide bottles that has been there for 50 years. The fact that plant science figured out in 1979 that a drugstore aspirin could activate a whole plant immune response, and that this research has been sitting in journals ever since, confirmed and reconfirmed, without ever reaching the seed packet or the garden center shelf, that is not your failure. That is what happens when knowledge has no commercial sponsor. Nobody needed you to know this, so you didn't. What you are going to know by the end of this video is the following. You will understand the specific biological system inside your pepper plant that makes this possible. The name of the master switch, how it works, and what happens inside the leaf when you activate it. You will understand the one mistake that 9 out of 10 gardeners make when they first hear about this method. The mistake that makes them conclude it does not work, when the real problem is timing. And you will have a complete protocol. The exact dose, the exact frequency, the exact time of day, the exact type of tablet, and the exact sequence from the day you transplant to the last spray of the season. The story of how this knowledge was found, and why it disappeared from public conversation so quickly, begins in the United Kingdom, in the year 1979, at a research institute called Rothamsted Research, which is one of the oldest agricultural science stations in the world. A plant virologist there named R.F. White was studying tobacco mosaic virus, the pathogen that destroys tobacco crops, and he had a question that at the time seemed almost absurd. What if a common aspirin tablet, dissolved in water and applied to the plant, could make the plant resistant to the virus? White's reasoning came from what was already known about plants under attack. When a plant is infected by a pathogen, it produces a chemical called salicylic acid at the site of infection. This was already documented. What nobody had tested was whether giving the plant salicylic acid from the outside, through the roots and leaves, from an aspirin dissolved in water, would trigger the same response. White published his results in the journal Virology in 1979. The answer was yes. Plants treated with dissolved aspirin developed resistance to tobacco mosaic virus. The resistance was real, measurable, and reproducible. He had found the switch. The research community took notice, but slowly, because there was no funding mechanism for this kind of finding. You cannot patent aspirin. You cannot build a product line around a tablet that costs 5 cents. The large agricultural companies had no reason to invest in promoting the finding, so they did not. White's paper sat in the archive, cited by other researchers, replicated in other labs, confirmed again and again, and never once mentioned in the commercial gardening world. Give this video a like for R.F. White. He did the work. He published it honestly. He proved it worked. And for over 40 years, the people who should have been telling gardeners about this finding were busy selling them something else instead. In 1990, Two researchers at Rutgers University, Jeanette Malamy and Ilya Raskin, published a paper in the journal Science that answered the next question. They proved that salicylic acid is not just a byproduct of infection. It is the plant's own internal hormone, responsible for a biological state called systemic acquired resistance, a whole plant immune condition that prepares every cell in the plant, from the root to the tip of the newest leaf, to fight off pathogens it has never even encountered yet. The discovery earned the kind of attention in the scientific community that rarely translates into garden advice, but it was the foundation of everything that came after. Then came the part of this story that should make you stop and read a sentence twice. In the early 1990s, a molecular geneticist named Joseph Riles was working at a company called Ciba Geigy, which is one of the world's largest manufacturers of commercial fungicides. Ciba Geigy funded research into salicylic acid and systemic acquired resistance. Riles and his team created plants that were genetically incapable of producing salicylic acid, plants that had no internal immunity signal at all. And they showed that these plants could not develop systemic acquired resistance under any conditions. They proved, definitively, that salicylic acid is the non-negotiable key to whole plant immunity. A fungicide company paid for the proof that aspirin activates a plant defense system that makes fungicide unnecessary. Ciba Geigy later became Novartis. The research was published. The conclusion was never advertised. The practical confirmation came in 2004, when a researcher at the University of Rhode Island, working with plant scientist Dr. Rebecca Brown, tested aspirin water spray on vegetable crops at garden scale, not in a controlled laboratory chamber, but in real garden beds, with real weather, real soil, and real disease pressure. The results confirmed what White had found in the lab 25 years earlier. The aspirin spray worked in the garden. Dr. Brown's work is publicly available and university endorsed. It did not start a product line. It did not generate a press release from any fungicide manufacturer. It added one more confirmation to a body of evidence that has been accumulating for nearly half a century. Evidence that you are only hearing about now because the economic incentive to keep it quiet has always been stronger than the economic incentive to share it. Now here is what is actually happening inside your pepper plant when a dissolved aspirin tablet soaks in through the leaves. Aspirin, acetyl salicylic acid, is converted by the plant's own enzymes into free salicylic acid. That salicylic acid moves to the plant's cellular fluid to reach a protein called NPR1, which functions as the master switch for the plant's immune system. When salicylic acid binds to NPR1, that protein unlocks a cascade of defense genes. Think of it as a security guard who has been sitting at a desk reading a newspaper. When the alarm sounds, he opens a cabinet, pulls out 40 different weapons, and stations them at every door. The alarm, in this case, is the salicylic acid from your aspirin tablet. The cabinet is the NPR1 protein. The weapons are a family of proteins called pathogenesis-related proteins, and some of them have one specific job: breaking down the cell walls of fungi. What makes this different from every bottle in the garden center aisle is the mechanism. Fungicides kill by direct chemical contact. They hit the pathogen from the outside. When you stop spraying, the plant has no more defense. The plant itself has done nothing, learned nothing, built nothing. Aspirin-induced systemic acquired resistance arms the plant from the inside. The plant produces proteins, specifically beta-1,3-glucanase and chitinase, that break down the structural component of fungal cell walls. The plant also produces peroxidases that strengthen its own cell walls against penetration. This immune state lasts approximately 20 days per application. And here is the detail that researchers confirmed in a study published in the European Journal of Plant Pathology in 2020. In sweet pepper specifically, salicylic acid applied before infection upregulates exactly these defense proteins, PR1, beta-1,3 glucanase, and peroxidase, and the plant becomes measurably resistant to Alternaria alternata, the black mold that wipes out late-season pepper crops. The research is peer-reviewed. The protection is real. What does this mean for your garden right now? It means that for 20 days after each application, your pepper plant is running its own immune defense, without any chemical purchase, without any spray residue on the fruit your family eats. There is one mistake that makes this method fail, and 9 out of 10 gardeners who hear about aspirin spray make it.
They hear about it in July, when their plants are already sick. They dissolve a tablet, they spray the diseased plant, and they wait for improvement. A week passes. The spots do not heal. The rot does not retreat. They conclude that aspirin spray is garden folklore that does not actually work, and they go back to the fungicide aisle. Look at what that mistake actually is, because the logic of it seems completely sound. You have a sick plant. You have learned that aspirin activates plant immunity. You spray the plant with aspirin. This looks exactly like the right action. The problem is timing and biology. Systemic acquired resistance takes days to build. The process requires the plant to manufacture new proteins and distribute them throughout its tissue. That manufacturing happens in healthy cells. Once you can see visible disease symptoms, the dark spot on the leaf, the soft ring on the stem, the blackening root, the cells in the infected area are already compromised. They are not producing defense proteins, they are responding to damage.
Spraying aspirin on those cells does nothing to the infection that is already there, because there are no healthy cells left in that area to arm. The spray does not cure. The spray prevents. And you cannot prevent something that has already happened. The gardener who sprays in July sees no result, puts the bottle away, and tells the neighbor that aspirin spray is a myth. The neighbor believes them. The knowledge gets one more strike against it, not because it failed, but because it was used in the wrong moment. You could not have known this on your own, because nobody watching a plant collapse in August has any visible reason to connect that collapse to the fact that they did not spray 6 weeks earlier in June, before any symptoms appeared. The cause and the effect are separated by the entire visible life of the problem. Write this in the comments. Did you ever spray something on a plant that was already sick, wait for it to work, and wonder why it failed? Tell me your region, too. I want to see how widespread this timing problem really is. Here is the full protocol, and every step matters. Step 1. Buy plain, uncoated aspirin tablets at the pharmacy. The standard tablet is 325 milligrams. Coated tablets and buffered tablets have a polymer layer that does not dissolve in water, and the active ingredient never fully releases. Uncoated, plain aspirin only. Step 2. Dissolve 1 tablet in 1 gallon of warm water, that is approximately 4 liters. You can add 1 teaspoon of mild dish soap to help the spray stick to the leaf surface. Do not add more than 1 tablet per gallon. Above a certain concentration, salicylic acid becomes toxic to the plant. You will see yellowing, brown burns, and leaf drop. 1 tablet, 1 gallon, every time. Step 3. Start 1 to 2 weeks after transplanting your peppers into the ground or their final containers, before any sign of disease appears. If you see spots when you start, you are already past the preventive window for those cells. Begin with healthy plants in healthy soil before the disease season begins. Step 4. Apply in early morning or late evening. Never spray in full midday sun. Water droplets on leaves under direct sun act as magnifying lenses and will burn the leaf tissue. Early morning gives the leaves time to dry before the hottest part of the day. Step 5. Spray until the leaves drip. Cover the top surface of every leaf and the underside. The underside of the leaf is where fungal spores land and germinate first. It is also where most gardeners forget to spray. Every leaf, top and bottom, wet until dripping. Step 6. Repeat every 2 to 3 weeks throughout the entire growing season. The immune state from each application lasts approximately 20 days, so a 2-week interval keeps the plant continuously protected with a small overlap. Stop early, and the protection ends. The plant returns to its baseline, unprimed state, with no defense advantage and no memory of the treatment. 8 applications across a 16-week season costs 40 cents and protects your pepper crop against Phytophthora root rot, Alternaria black mold, grey mold, and bacterial wilt. The 4 diseases that account for the majority of pepper losses in home gardens. The fungicide path runs $30 to $40, requires constant reapplication, leaves chemical residue on fruit, and does not build any defense in the plant itself.
A plant that has been on the aspirin protocol for 6 weeks has been manufacturing its own antifungal proteins continuously since the 2nd week after transplant. Proteins that break down fungal cell walls at the cellular level before any visible infection can take hold. The research confirming this is not new. It is not experimental. It has been peer-reviewed and reconfirmed across multiple decades, multiple countries, multiple research institutions. The paper that started it was published 46 years ago. The company that proved the mechanism was the same company that sells the product you were supposed to buy instead. Your peppers can protect themselves. They have been capable of it all along. What they needed was the signal. That signal costs 5 cents and dissolves in a gallon of water. And here is the detail that will change the way you think about every seed you plant next season. The immune memory that aspirin builds in your pepper plant may not end with the plant itself, because researchers studying epigenetic inheritance in plants have found evidence that salicylic acid treatment can influence the seeds the plant produces, so that the next generation carries a measurable head start on disease resistance before it ever meets a pathogen. Think about what that means for a moment, and let it settle. You spent 40 cents this season and protected your crop, and the seeds you save from your healthiest peppers in September may carry something forward. Not a guarantee, not a miracle, but a biological echo of the immune state you triggered, passed through the seed coat into the next generation's first cells. That is not the behavior of a chemical spray. Chemicals do not pass anything forward. They dissolve, they wash away, and they leave the plant exactly as vulnerable as it was before the bottle opened. What you built this season with dissolved aspirin tablets is a relationship between you and the plant's own biology. And relationships have a way of continuing. Now step back and look at what actually happened across the whole span of what you have learned, and let the pieces fall into a single picture. You have a plant that already carries the machinery for whole-body immunity. You have a signal molecule, salicylic acid, that is the key to unlocking that machinery. You have a master switch inside the plant called NPR1 that, when activated, opens a cascade of defense proteins covering every cell from root tip to newest leaf. And you have a drugstore tablet that delivers that signal for 5 cents. Every mechanism Writer 1 walked you through, the proteins, the cell wall breakdown, the peroxidases, the 20-day immune window, all of it is one system. Call it what it is. The plant's immune layer, dormant until signaled, and fully capable of handling the pathogens that have been costing you money and heartbreak every summer. But here is the new angle that the biology alone does not give you, and this is the one you will carry out of this video.
Salicylic acid at the dose you are dissolving in a gallon of water has zero direct antifungal activity in laboratory conditions against Phytophthora capsicii, the pathogen that causes crown and root rot, the one that killed your plant in 8 days. Zero. The tablet does not touch the fungus. It never reaches the fungus. What happens instead is that the plant itself produces the proteins that break down the fungal cell wall, and the fungus meets the plant's immune response before it can penetrate deep enough to do damage. That means everything depends on the plant being ready before the encounter. It means the immune state must be running before the spore lands on the leaf, before the water carries the pathogen to the crown, before any of the biology of infection begins. The tablet is not a weapon. The tablet is a wake-up call. And a wake-up call only works if you make it before the trouble arrives. That single understanding changes everything about how you garden. You are not buying a cure. You are building a plant. And a plant that has been building its own defense proteins continuously since the second week after transplant is a fundamentally different thing than a plant waiting to be rescued by a bottle. Walk out to your garden and picture 2 plots side by side, because this is exactly what the choice looks like in practice. Both beds have the same variety of pepper plant. Both were transplanted on the same day from the same seedling tray. Both got the same soil amendment, the same watering schedule, the same mulch. The gardener in the first bed reached for the fungicide spray bottle in the 3rd week of June because the neighbor had always used it and the label promised protection. The gardener in the second bed dissolved 1 plain aspirin tablet in 1 gallon of water, added a splash of dish soap, and sprayed every leaf, top and bottom, in the early morning while the dew was still on the grass. By mid-July, both gardens looked similar from a distance. Then came a wet week in late July, the kind of week where overnight humidity sits at 90% and the soil never fully dries between rains. The garden center gardener came out on a Friday morning and found 3 plants with dark rings at the crown and soft spots on the lowest leaves. By the following Friday, those 3 plants were on the compost pile. The aspirin gardener came out that same Friday morning and found the same humid conditions, the same mud, the same pressure, and no dark rings. The plants held. They have been holding for 20 days before that rain ever arrived. What was the one difference that was invisible from across the yard? One of those gardeners was spraying the outside of the plant and hoping the pathogen never got through. The other was building the inside of the plant and trusting the immune layer that was already running. The difference was not in the product. The difference was in understanding which direction the protection comes from. And here is where the chain reaction becomes visible. Because aspirin spray does not solve one problem at once. It closes 3 at the same time. The first problem it solves is disease protection, which you now understand completely. The second problem it solves is chemical residue on your fruit, because the plant is doing its own work and you are not adding synthetic fungicide to the surface of a pepper your grandchildren are going to eat off the vine in August. The third problem it solves is cost. 40 cents versus $40, and the $40 path requires you to keep buying every season, requires you to keep reapplying every 7 to 10 days, and still leaves you with a plant that has built nothing, learned nothing, and will be just as defenseless the first week you miss a spray. 1 action, 1 tablet dissolved in 1 gallon of water, applied on schedule from transplant day forward, closes the disease problem, the residue problem, and the budget problem at the same time. 1 purchase, 40 cents, 16 weeks of protection, and a plant that is working for you instead of waiting for you. So now you are standing at a choice, and I want you to think about it clearly and then tell me which path you are on. Path A is the path you have been on, and it looks like this. You buy 2 or 3 bottles of fungicide this season. You spend somewhere between $30 and $40. You spray on a 7 to 10 day schedule through the whole summer. You rinse the fruit before it comes inside. And in a wet year, you still lose plants because the chemical could not reach the infection that had already moved underground. You have paid for surface protection on a problem that lives beneath the surface. Path B is this. You walk to the pharmacy. You spend less than $1 on a strip of plain aspirin tablets. You dissolve 1 tablet in 1 gallon of water every 2 to 3 weeks from the week after transplant. And your pepper plant spends the growing season running an immune system that is already inside it, already capable, already designed for exactly this situation. And you have fruit without residue and a garden budget that kept $40 in your pocket. Write A or B in the comments, and then write why. Not for me, but for yourself. Because when you say the reason out loud, you hear something you could not see before. And while you are down there in the comments, tell me how many pepper plants you grow and what region you are in, because I want to see where this timing problem has been hitting the hardest and whether there are regional patterns in when people first notice the disease pressure each season. Now let me show you something that is happening inside your pepper plant right now, whether you have treated it or not. Down in the cells at the base of the stem, where the plant meets the soil and the root system begins its work underground, there is a quiet chemical conversation happening at every hour of every growing day. The plant is reading its environment through chemical signals, sensing humidity, sensing light, sensing the composition of the soil water that moves through its roots. It is doing this the same way it has been doing it for millions of years, long before there were garden centers or fungicide bottles or YouTube channels about growing peppers. The machinery for immunity is there. The proteins are waiting. The master switch is present in every cell. What the plant is waiting for is the signal that tells it the threat is real and the defense should begin.
When you give the plant that signal, when you dissolve the tablet, mix the water and spend 15 minutes in the early morning moving through your garden with a spray bottle, covering every leaf from top and bottom until the water runs off, you do more than just prevent disease. You restore a relationship between you and the plant that the chemical approach broke. The chemical approach said, "I do not trust you to protect yourself, so I will coat you in something that kills what comes near you, and we will hope that is enough." The aspirin approach says something different. It says, "I know what you are capable of. Here is the signal. Do what you already know how to do." What returns to the plant when you do this is its own biological purpose, the full expression of what it was built to be, producing fruit, resisting disease, building immunity, growing through the season without depending on a product that costs $40 and leaves residue on the peppers your family eats. The plant was never helpless. It was never waiting for a spray bottle to save it. It was waiting for the conversation to begin. The power is not in the chemical bottle on the garden center shelf. The power is in knowing what signal the plant has been waiting for and giving it before the season turns against you. So take this with you when you walk out to your plants tomorrow morning. Walk out early when the ground is still cool and the light is still low, and look at what you have growing. Every one of those pepper plants is running a biological system more sophisticated than anything in the garden aisle. And now you know the name of the switch and the cost of the key and the timing that makes all the difference. If this reached you in time, share it with the gardener next door who is still buying bottles, because the knowledge only spreads if someone carries it forward, and you are the person who now has it. Go deep.
Everything is down there.